Hey, welcome everybody. My name is Ruth Kapelis. I'm the National Content and Media Relations Manager, and on behalf of JDRF, I'd like to welcome you tonight to our Let's Talk T1D educational series. We're very excited to present you with more information on cell therapy for T1D cures. The presentation will be approximately 30 minutes, and then we'll open up the session to your questions to our presenter. If you've been here before, thank you for coming back. If this is your first time, JDRF is a global leader working towards an end to type 1 diabetes through both research, funding, and advocacy. We also support people and families in the T1D community from the time of diagnosis by helping those living with T1D to have better, healthier, and safer lives while we're working to find cures. I'd also like to begin by respectfully acknowledging that the JDRF Canada National Support Office is located on the traditional lands of the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. The education series offers information on a new topic approximately every other month. Our final one for 2024 will be T1D and self-management, including device fatigue. We also host a virtual connection series on the fourth Wednesday of each month. This is an unstructured opportunity to connect with others with no formal programming. And the next connection series will be taking place on November 27th at 12 p.m. Pacific time and 7 p.m. Eastern Standard Time. Just a reminder once more that we're a really big group, so please do your best to minimize background noise. We will have a chat where you can post your questions, and we will put contact information in that chat as well if you have questions you want to ask us after this meeting. And now I'd like to begin the presentation. So curing T1D is the primary goal of JDRF, and obviously most of the, if not all of the 300,000 Canadians living with T1D and their families. Cell therapy is one of the most active and fast-moving avenues of research in this area, but what is it and how close are these approaches to becoming a reality? We're very excited to be joined tonight by Stephen Paraskevas. He's got a very long title, MD, PhD, transplant surgeon, director of pancreas and islet transplantation, and director of transplant research, and professor, Department of Surgery, Faculty of Medicine and Health Sciences at McGill University. And I'm proud of myself for getting through that and very excited to turn it over to you now for the presentation. Thank you.
Thank you very much, Ruth, and hello and good evening, and thank you all for your time in attending this webinar tonight. As Ruth just mentioned, I'm a transplant surgeon at McGill University Health Centre in Montreal, where I'm also responsible for the pancreas and islet transplant program. I'm involved in several clinical trials of cell therapeutics related to type 1 diabetes. I'm very grateful to JDRF Canada for hosting this event and for supporting the mission of many researchers and scientists throughout the country who share a goal of developing novel treatments and a cure for type 1 diabetes. So with that, I'm going to just give you a short outline of what I'll be talking to you about tonight. I'm going to touch on several topics, beginning with the basics of cell therapy and the obstacles of the immune response to any transplanted cell. We'll touch on the current standard of islet transplantation, its results, the risks and benefits, because they apply equally to the newer therapies that are coming on the horizon. I'll talk, of course, about the advent of stem cell-generated islets, the current Vertex trial, its interim results, and then some preliminary data about trials involving engineered devices and some new approaches to protect transplanted cells from the immune response. And finally, I'll discuss some interesting recent publications on the ability to generate personalized islets from an individual's own stem cells and how this might help people with diabetes. So let me just review a few basic concepts, and I know they may be well known to most people in the audience, but just in case. The pancreas, of course, is an organ in the back of the upper abdomen, which has a dual role related to digestion and metabolism of nutrients. Of course, 99% of the organ is glandular tissue, which makes digestive enzymes that allow us to break down the food we eat. And dispersed within this tissue are little microorgans called the islets of Langerhans, and they're tiny clusters of cells which make hormones, mainly insulin and glucagon, that allow us to control the level of glucose in our blood. The cells in the islet that make and secrete insulin in particular are called the beta cells. And in various tissues of our body, insulin, which is released into the blood, acts on insulin receptors like a key in a lock. And the action of this lock is to enable glucose to enter the cell where it is needed for energy and for life. Now, our immune system has evolved to protect us mainly from bacteria and viruses that might invade the body and threaten our health. It does this by being able to distinguish our own proteins and our own molecular markers from those which are not our own, which it reacts to, assuming that they are intruders. People with type 1 diabetes, the immune system misfires and mistakenly responds to proteins on the beta cell, resulting in an immune attack by various cells of the immune system, shown here in the lower right, ultimately causing the destruction of all the beta cells in the pancreas. Unable to produce their own insulin, people with type 1 diabetes have to then rely on injections of the hormone in order to maintain normal blood sugar and be able to use sugar, which is, as I said, essential for life. Now, as many of you also must know, taking insulin injections is an imperfect way to control blood sugar. Despite the best efforts of many people, blood sugar fluctuates and can fluctuate quite a bit, causing damage to other tissues. One way to enable a person to make their own insulin is called islet transplantation. And this is
Dr. Paraskevas, sorry, could you please click again on your slide because it's kind of hazy again.
Oh, how's that?
Sorry, it went, no, there, perfect.
Okay, that one seems to be giving us problems tonight.
Yeah.
So islet transplantation is the first and most widely administered form of cell therapy for diabetes. And in islet transplantation, the pancreas of a deceased donor, you see on the left of that diagram, is recovered in the same way that we recover kidneys, livers, lungs, and heart for transplantation. And then we take the pancreas to a special lab, such as we have at the MUHC, where the connective tissue in the pancreas is broken down by a special mix of enzymes. The acinar cells, which are the ones that produce digestive enzymes, and the islets are separated from each other because the islets are lighter and the acinar tissue is heavier in density. And taking advantage of this difference in density, we're able to produce basically a purified preparation of only the islets, which amounts to about 3 or 4
Sorry, we
cc's.
can't see that slide. It hasn't turned over.
Can't see anything.
Okay, how about I'll go on to the next one. How's this?
Perfect, that's good.
Okay, so we're able to create this purified preparation of islets, which amounts to a few cc's of tissue.
I guess I just went and left me recording.
Sorry, could you please go on mute? Sorry, there, I muted. Good. Sorry, Dr. Paraskevas, go ahead.
And the islets are injected into the liver of the recipient. You know, most people ask, why the liver? Why don't we put them in the pancreas? The liver has a dual circulation. It has an extra venous circulation called the portal system, through which it processes nutrients. And because of this, it's able to tolerate what is essentially tissue being put into vascular space, where it's not supposed to be. And it doesn't damage the liver. And the islets, most of them appear to survive there and start producing their insulin and glucagon there. When we try to put the islets anywhere else, like in the pancreas, we just cause damage to the organ, and we don't get the desired response, because it's never really been done in humans, because many years ago these studies were done to demonstrate that islets couldn't be put almost anywhere else except the liver. So that's where it is done as a standard today. So here are some photos illustrating the process of islet isolation. In photo A at the top left, you see the pancreas. I hope no one's squeamish when they look at it, and the surrounding tissues that it comes with when we remove it from the donor. You see the spleen on the right of the pancreas, and the duodenum is a little piece of small bowel on the left. In photo B, those extra tissues have been removed, and we just have the gland itself. In C, the pancreas is being injected with a very special enzyme solution that breaks down the connective tissue. In D, we have a circuit attached to a bioreactor called the Ricordi chamber, where the dissociation of the pancreas takes place at 37 degrees. In E, we see dispersed pancreatic tissue that we get out of the Ricordi chamber, and we use a special stain which turns the islets dark red, and you see those in a few of those in that picture, and the acinar tissue looks orange. In F, we have the total tissue produced by the dissociation. We may get 60 or 70 cc's of tissue out of a pancreas. And then it's placed in the special centrifuge called a Cobe cell processor in G, which separates the tissues on the basis of their density, and we get H, photo H there, a very relatively pure preparation of just the islet tissue. Here is our team working in a special lab under very, very environmentally controlled conditions, where we produce these cells and where they're tested and evaluated before being released to a patient. And here we are in the radiology suite at the MUHC at the Royal Victoria Hospital, where one of our interventional radiologists here is Dr. Gallix, performing an islet transplant. On the screen in front of him, you can see the vessels of the patient's liver, highlighted in dark gray there, as the small catheter is inserted into the portal vein through the skin. The patient's awake with just a little freezing on their right side, and the procedure takes about 45 minutes. I'll try to play you this video, which shows the islets going in. It's always pretty exciting to see. They're like tiny little grains of sand, and if you look, well, it's a little choppy, but if you can see that they're running into the liver there. But before we get to this step, the lab staff have already subjected them to a huge battery of tests to make sure they're safe, they're alive, they're actively making insulin. It's essentially 3 days, 3 long days of work to produce a batch of islets like this. Now, one issue is that islets definitely go into a hostile environment in the recipient. Just to review, in the context of type 1 diabetes, abnormal autoreactive immune cells, such as this T cell here, which should have been deleted during our development. You can see it there, equipped with all sorts of receptors and loaded with granules of lethal proteins and cytokines that kill the cells it identifies, react against normal self-proteins on the surface of the beta cell. And this causes an attack that can kill the beta cell. In the transplanted cell context, the beta cells from one individual that we transplant into another, the proteins on those cells look different to the recipient's immune system, and these non-self proteins trigger normal, healthy immune cells whose main job it is to try and root out and kill bacteria and viruses. But because these non-self proteins look different, these, what we call alloreactive immune cells, attack the transplanted beta cells. And this process, which can happen in any transplanted cell or organ, is what we call rejection. In order to curb rejection, we give the recipient special medications that inhibit these normal alloreactive cells and prevent the graft from being attacked. Now, anti-rejection medications are good to help the graft survive, but they have some unwanted side effects. For example, they make our immune system weaker in general, so it doesn't respond to a vaccine like it should. Because of the need for anti-rejection medications and their side effects, the decision to give someone an islet transplant involves a careful risk-benefit analysis. So in general, the people who stand to benefit from islet transplantation either have severe glucose control problems or have lost the sensation or awareness of hypoglycemia and can have dangerously low sugars without knowing it, or who are already taking anti-rejection medications, such as people who had, for example, a kidney transplant because they lost kidney function due to their diabetes. The benefit, however, is becoming more and more clear. And it's not an insignificant benefit. We can take someone like our pre-transplant patient on the left, who is in the normal range of sugars only 16% of the time, and turn them into someone like our post-transplant patient on the right, who's in the normal range 99% of the time. And that has a significant and positive impact on their global health. Also, some good news from the team in Edmonton. This publication from a couple of years ago shows that transplanted islets can function for up to 20 years. In this report, the Edmonton group pointed out that some patients had stable graft function, lasted a long time. That's the group in orange. And some patients had sort of lost graft function over time, and that's the group in green. Here we see in the top panel fasting C-peptide, which is a protein that's produced during insulin production, being quite normal and stable over the course of many years in the orange group. Similarly, they enjoyed a near normal and nearly stable beta-2 score, which is in the middle panel. The beta-2 score measures the strength of beta cell function. And in the bottom panel, we see their total insulin use as a percentage of pre-transplant total. So most of the orange group stayed close to about 10 to 20% of their pre-transplant insulin use, indicating how much better their control is compared to before. I should say that initially, at least half the recipients come off insulin completely. And over time, as you can see, it slightly increases, meaning that people may take 2, 4, 5 units of insulin and end up with that 99% time in range, which is what we're really looking for to keep them healthy. The orange group also enjoyed normal hemoglobin A1C as opposed to those who lost their graft function. And that's in the top panel there. And ultimately, the A1C is our measure of glucose stability. And that's really the key aspect to the improvement of health. And in the bottom pane is the fasting plasma glucose, also an important measure of health. And that you can see is also staying very stable and low in the orange group. So while islet transplantation clearly works, there are important limitations. First of all, the reliance on organ donors is problematic. Relatively few organ donors qualify to donate their pancreas, and the procedure to purify the islets is difficult and costly. Often one injection of islets from one donor is not enough, and so it can take 2 or 3 different injections to stabilize a person's sugars. And of course, as I mentioned before, the need for anti-rejection medications is another important limitation. But we are now witnessing a development that may change the field permanently. Dr. Douglas Melton, a Harvard researcher in developmental biology, and it should be known, a father of 2 children with type 1 diabetes, devoted nearly 20 years of his career to what is essentially represented in this diagram in this paper. This is the reproducible protocol for the scalable, large-scale maturation of beta cells from pluripotent stem cells. And stem cells are a natural population of cells in all of us that retain the genetic potential to become any type of tissue in our body. So this process, which the Melton lab developed laboriously over decades of research, turns on and off particular genes in a precise sequence and recapitulates the way beta cells develop naturally during gestational development. And the result is mature cells with surface markers of natural beta cells that respond to glucose, manufacture and package insulin the appropriate way in secretory granules, and release it in quantities comparable to natural cells in response to multiple sequential glucose challenges. Just what we expect from an adult beta cell, and available in virtually unlimited quantities. So here's where we find ourselves in 2024. We now have we now are on the doorstep of artificially generated beta cells functioning normally in human recipients. The cell source challenge appears to have been answered. We still have the issue of protecting these cells from immune attack. Remember, the stem cells used by the companies that are trialing these products now are developed from human donors. They will be perceived by any recipient's immune system as non-self and subject to allograft rejection. So even these cells require anti-rejection in order to survive in their recipients. The focus of much research now is to find a way to protect them from immune attack. So there are several ways of doing this. One potential way is by making a physical barrier that protects the implanted cells from the immune system, while at the same time allowing nutrients and oxygen in to keep them alive, and allowing insulin produced by the cells to leave. The engineering of such materials is very, very precise in this regard, and this approach is called encapsulation in the upper left. Other approaches use scaffolding to anchor cells down and allow them to be exposed to a protective local environment of signals that deter the immune response.
Sorry, the slides are wonky again. If you could click on your slide one more time, because we can't see the scaffolding or encapsulation. There's okay. 2 in 1.
Oops, how's that?
Yeah, you need to go back one now. I think now we're at the forward track. Perfect. Now it's perfect.
Okay. Sorry, it's a little slower at your end, I guess, than in mine. Okay, so scaffolding allows, as I mentioned, the cells to be exposed locally to either medications or signals that decrease the immune response to them in a very controlled way. And gene editing on the lower right is the prospect of engineering cells not to express any self proteins or non-self proteins that can cause an immune response in the first place. And so research shows that these cells can actually still function normally while being essentially invisible to the immune system. So gene editing is another important technology that is available to date to modify cells. Perhaps they're not even detected by the immune system. I assume the slides are advancing now?
Yeah.
Okay. So the most advanced trial of stem cell generated islets is by Vertex. Some of you may know this one as the VX-880 trial. It is now called the Forward trial. It's enrolling patients with type 1 diabetes and severe recurrent hypoglycemic events. The stem cell generated islets are transplanted in an identical fashion to deceased donor islets into the liver, in other words, and with recipient taking standard anti-rejection medications. This is data that I'm showing you here is from the Forward studies, all publicly available, and it was presented at the American Diabetes Association meeting in June. So there's nothing here that's confidential. At that point in June, 12 participants had received these islets and all showed excellent function. And 11 of the 12 showed at least a reduction in insulin injection requirements over the first few months after transplant. And 7 of 10 who had completed 180 days post-transplant were no longer using any insulin. This is their CGM data, starting at baseline in the column on the left, day 90, 180, 270, and the end of 1 year. And you can see that function continues to improve over the course of the first 12 months, from about 50% time in range to almost 96% time in range. But it should be noted, of course, as I mentioned, these patients are all still taking anti-rejection, and this comes with the risks and potential hazards that have always been associated with these medications, are nonetheless used in thousands of organ and tissue and cell recipients around the world. The next step, that of protecting the islets so that no anti-rejection is needed, is the focus of the next Vertex trial, initially called 264 and now known as Upward. In this trial, the stem cell generated islets will be placed in an engineered device and implanted into the abdominal wall of a participant. The device is made so as to keep immune cells out while allowing oxygen and nutrients to come in and glucagon and insulin to be able to leave. So very, very precise parameters that are required. The material engineering of these devices is incredible. The trial will assess the safety and tolerability of these devices mainly. Participants will receive no anti-rejection medications. So as you can see, the sample size is approximately 23 subjects. The primary outcome is safety and tolerability. This is an early phase study. So they want to make sure that there are no complications of implanting and having the device. And secondarily, they will be looking for example, hemoglobin A1C changes, CGM metrics, or even insulin independence. But the primary goal of the study is to make sure that this device is safe. Other companies have started trials of their own. Cernova, here a Canadian company, has developed a scaffolding type of device which may hold immune modulating drugs in the future. They first wanted to test the ability again of the islets to survive in the device, which is implanted only under the skin, so not so very easily done. These patients received deceased donor islets in the standard fashion in the liver and with anti-rejection medications, but a small amount of the islets were placed in what is called the cell pouch made by Cernova. All participants had normal function, so they had good quality islets. And in some from whom the device was removed, living islets were found in the device. So this suggests the device is capable of supporting implanted cells and keeping them alive. The immune modulation strategy is obviously what we look forward to next. But as I show you here, encapsulation, another approach to protecting the cells, doesn't have to necessarily imply large devices with the problem of providing enough oxygen diffusion to keep the cells alive. For years, an approach called microencapsulation, which is shown in the middle there, was very popular, and that involved making small bubbles of a material called alginate that would each contain a single islet. Today, this principle is further refined in a process called conformal coating on the right. This covers the cells in a very thin layer of material that prevents immune cells from attaching to them. It's so thin that nutrient and oxygen diffusion isn't an issue. And in this study, which is an experimental study in animal recipients, conformally coated islet cells showed normal insulin content. They're very much alive, as you can see on the photo on lower left. And they make insulin and contain an appropriate amount of insulin, meaning they're getting nutrients that they need to stay healthy. But now we've got some newer solutions that may yet make encapsulation obsolete. And In another true first, group in China recently reported that they took fat cells from a woman with type 1 diabetes who had previously had and continued to have a liver transplant, was living with a liver transplant. They isolated the stem cells from the fat sample and differentiated them into islets and transplanted the islets into the woman's abdominal wall. She demonstrated robust islet function with insulin requirements dropping to zero fast, as you can see in the top there, the insulin requirements going to zero by day 90. Fasting glucose returning to normal in panel B. And her time in range going up to about 98%, as you can see in panel C. Her A1C at the bottom stabilized at a very normal level. This person was and is on anti-rejection medications for a liver transplant. But what is really exciting about this is the ability to produce individualized islets for someone from their own tissues. However, as I've explained already before, these are self-protein expressing cells, and they could be attacked by autoreactive immune cells. And we just don't know what would happen to them if the person was not on anti-rejection. We suspect that they may not survive in the long run. We don't actually know what will happen to them in any case, since anti-rejection medications don't traditionally work as well against autoreactive immunity, and over time, autoimmunity can still be an issue even in transplanted organs and tissues. But this is a huge step forward, there's no question. So here we are, a very exciting time for the treatment options for people suffering with type 1 diabetes. We have deceased donor islets and now becoming widely available in Canada and showing evidence of functional stability extending out to many years. That's on the left. This option comes with known risks of anti-rejection, but is otherwise safe and can be quite effective. It's limited by the availability of donor tissue. You have in the middle the advent of stem cell generated islets, which can be done as a standard transplant using anti-rejection. This innovation just by itself will vastly increase the availability of islet transplantation by eliminating the constraint of donor tissue. We don't yet know how much it will cost, and this may affect the rate at which it's adopted by public health systems like Canada's. But barring any major problem, this is probably going to be available in the 5 to 10 year time frame. Then on the right, you have stem cell islets generated from autologous tissues. This is still in very early stages, but the report that I showed you is very promising. This will deliver essentially self-islets to an individual and could be a great advance for people with non-immune forms of diabetes, whose islets may be dysfunctional or not producing enough insulin. For type 1 diabetics, something will have to be done to protect them from the immune system, either with engineered devices that are able to maintain living cells inside them, or by further modifying the cells generated to edit out completely the expression of self-proteins that stimulate an immune response. This way, effectively, you wouldn't even need autologous cells. You would have a product of generic cells that would not be rejected in any way, either by autoimmune or by alloimmune cells. It's not so far-fetched as it sounds. There are already investigators working on this in many places, including some in Canada. So exciting times indeed. That's about it for this evening. I thank you again for your time, and I'm delighted to answer any questions you may have.
Thank you so much for that. I see a question here from Lily, as she asks, has anyone transplanted a patient's own umbilical cord blood cells, or is there a risk of autoimmune attack from that as well?
So thank you for the question. That is essentially what was done in the report from China. They are not the umbilical cord cells, slightly different type of stem cells than the ones we get from tissues like fat tissue that was used in that one. But the end result is that they were able to produce basically a supply of the patient's own islets. So in any case, this person, and they chose this person for a reason, that she had a liver transplant, was on anti-rejection medications. The purpose of this report was just to show that it can be done and that these cells will function. But clearly, the reason that patient was chosen for that report was because they're taking the medications already. If you gave these cells, if you took a person and made their own islets and gave them cells without any medication, the thinking is that we'd still have a problem unless we can protect or modify the cells so that it doesn't happen.
Oh, there's a lot of questions coming in now. I think there's one asking if children can receive stem cell transplants or this type of therapy.
Currently, there are 4 programs performing islet transplant in Canada. So let's start with the most standard procedure. And as I mentioned to you, it's still kind of a risk-benefit analysis with each individual. For someone who is very stable and has no complications of their diabetes, as often children do, the risks of taking anti-rejection at that point in their life probably outweigh the benefits. Our candidates for islet transplant, some of them are quite young adults, have all developed some condition which makes the risk benefit in favor of doing the transplant. Either they have trouble with poor control, hypoglycemias, or usually that is the main reason, or they've developed kidney problems and require a kidney transplant. So it's all relative to the issues that the patient or the person, pardon me, is facing. It will probably be the same for what are coming in through these trials, and of course, as a protective measure, most trials that we see these days enroll adults only. It's not that that people don't want to have children benefit from this, but in order to protect people from an unforeseen complication, most clinical trials are currently just enrolling adults. But islet transplants, which are a standard of care, they're not in trial anymore, are generally available to adults who have some compelling complication.
Sean asks about Sana biotechnology trial in Sweden that's using gene-edited cells. And he says the goal is to see if cells survive on MRI and if C-peptide is produced.
So there's a lot of interest now using, that's a great question, a lot of interest in how we can use CRISPR technology to change these cells. So if we don't do anything and we just have mature islets, they stand a risk of being damaged by the immune system. So there are a number of researchers around the world really looking at ways, and actually a few companies looking at ways to modify the cells so that they're less visible or invisible to the immune system. And even investigators in Canada, Dr. Christina Nostro in Toronto, and others who have looked at removing the molecules that cause the immune response, they're called HLA molecules, and thereby having a cell that doesn't stimulate the immune system, but is still perfectly functional. And that's very exciting because it suggests that no matter the origin of the cell, whether it comes from someone else or from a person's own tissues, that it will not be detectable by their immune system.
Sorry, a whole bunch of questions came in while you were talking. Connie and Jean are asking if they're trying to modify patients' own stem cells that were converted to beta cells and trying to modify the exterior markers on these cells so that their immune system doesn't recognize and attack them.
I'm sure someone's working on it. That report I showed you is very, very new. I'm certain that is the next step. And in my opinion, as someone who's in the field and applying these treatments to to people who need them, I would say that that would be the most exciting upcoming development. I don't know, how and when that will come to early phase trials, but all the tools are there to be able to do that. So hopefully soon.
There's a few questions that have come in, and I can see that my colleague Lara is answering them, just about sort of simultaneous studies regarding autoimmunity. And if, I know that screening and prevention is a big part of what JDRF helps fund as a pathway to a cure as well. But is that being done in any way in conjunction with cell therapy? Or are they two separate fields of study?
Could you just repeat that? Because I is that the question that I'm looking at here, or another one?
There's a few questions about the autoimmunity and the if there's a way to, obviously, to turn that off in someone without needing any kind of suppression.
Yeah, there certainly are ways of engineering treatments that kind of identify and destroy the cells that are autoreactive. I'm not sure any of them have gotten to more advanced trials, but that is a possibility. Someone here I'm looking at the chat has also mentioned teplizumab. So yeah, there are ways of decreasing or protecting the transplanted cells from the autoreactive cells. Of course, when someone with type 1 has already lost their cells, so they would need a transplant, a replacement of their capacity to make insulin, and then a way of protecting those cells from the immune system. The ideal, of course, would be if some of these immune therapies like teplizumab become so effective that we can give them to people who are just diagnosed, to kids who are just diagnosed, and in that early phase where they still have natural beta cells in their pancreas. And if we can arrest the process at that time, then you really are talking about a cure.
Sarah was asking, is there a specific time and range percentage that makes you a better candidate or better qualified for participating in one of these trials?
In general, I would say most of our recipient candidates are unable to achieve 70% time in range. We work as a team. I'm a transplant surgeon. We have Dr. Sarah Meltzer as our endocrinologist. She takes a look at all of these candidates from the perspective of have we optimized exogenous insulin, and so we try and see that if someone can make obvious changes to their insulin regimen and improve their quality of control, well, we'd rather that they do that first, and then if that doesn't work, and if they're unable to, say, achieve something like 70% time in range despite all efforts, then we can say, well, now you have potential to improve on that. But obviously, if you have 90% or if you have higher than 70% time in range and you're doing quite well with that, again, as I said, the risk-benefit analysis may not favor going on medications to get a few extra percent.
We had a question from Monica about the VX-264 upward trial, the one without the anti-rejection drugs. Is it in fact only for people who have AB or A blood types? And if so, are they going to be expanding that criteria?
I'm sure there's an effort to develop O or generic blood group compatible cells that Vertex. I'm not privy to that information, clearly the first cell line that was obtained by Vertex to develop into transplantable islets happened to be from a person with blood group A, and that has, of course, made the requirement for this trial that people are compatible with that person's cells, so they have to be A or AB. So yes, unfortunately, that is the case, but it really is a matter of, once we are sure that these products are working well and that the trials have achieved their goals. I don't work for Vertex. I'm certain that would be the next thing on their schedule, is to produce one that's available to all blood groups.
And the next question is another Vertex related question. So I understand you can only answer within what you can answer, but Kathy is asking if VX-880 is successful, but VX-264 is not. Would one of the other encapsulation devices work with the Vertex cells, and are they still working with CRISPR?
They're still working. I don't know the details of it, and so we will have to wait and see. I mean, the 2.6 upward is, getting started. I imagine by next year we'll have some preliminary data, and everyone's holding their breath. See what happens. I think it's a very exciting time.
This question here from Dan is: type 1 individuals appear to have dysfunction of the complete islet, and we hear little of these other cell types, alpha, beta, delta, epsilon, the Greek alphabet. Would a true functional cure require these other cells that also contribute to the body's response to glucose fluctuations?
Well, that's a great question. Of course, and interestingly, the process that Vertex uses to create mature islets, they're actually creating mature islets with all cell types in them. So the islets they produce look like the kind that we get from organ donors. They have all the cell types in them, and so it's a true replacement. What is the role of the minor cell types in the function of the beta cell? I think I'll admit that I'm probably not an expert in that, but to say that, of course, what diabetics lack are primarily beta cells. Yes, some diabetics have very, very acellular islets in their pancreas. After some time, they don't produce glucagon either. We don't know how that is related to the autoimmune attack. Clearly, it is. But yeah, the interesting thing about the Vertex islets is they're complete. They have all the cell types in them.
I think you've somewhat answered this, but we are still getting some questions about if you're producing some insulin still, is it worth it to augment it with this type of therapy?
Well, because we still don't get the ideal function out of, say, a deceased donor islet transplant. Sometimes we have to do 2 or 3 injections. We really kind of look for people who are insulin sensitive. That means that they produce none or close to none of their own, and that even optimally they use only about, the ideal kind of rule of thumb that we have is half a unit per kilo per day. So somebody who, say, is 70 kilos would be taking 35 units approximately or less. So we have done more than that, but pardon me, that's kind of our rule of thumb. We want people who are a little bit more on the insulin sensitive side, because we're giving them a graft that may not be quite as robust as we would want it to be. And also in the trials, which are very, very strictly controlled, they're insisting on people who have zero insulin production on their own. Somebody produces insulin and maybe doesn't has diabetes because their insulin production is insufficient. That's a little bit trickier with islets. Of course, it is an area where pancreas transplantation is still effective and available. It's less commonly done. But yeah, we get into a bit of a gray zone there.
I'm going to combine a couple of questions because there's some questions about the risk of the anti-rejection medications, and then are there other risks of doing this type of procedure other than the anti-rejection medication?
Okay, a great question. So anti-rejection, of course, it's a necessity. As I mentioned, it's the same medications that are given to anyone who has an organ transplant, for example. But if I needed a heart or a liver, I pretty much have no choice. I don't. I absolutely need that, and I have to. And justifying those risks is fairly easy. In islet transplantation, because there's an alternative, whether it's a good alternative is of course debatable. Then we get into the risk-benefit analysis. We have to look very carefully at the consequences of taking these medications. Primarily, anti-rejection does what it's supposed to do in reducing the effectiveness of your immune system. It's protecting the graft in order to do that. But what the result is that we're more susceptible to infections. Some people are susceptible to getting infections that they don't get at all when they don't take these medications, fungal and viral infections that our immune system takes care of without us even noticing. And things like our response to vaccines gets blunted. So while all our transplant patients still get vaccinated, for example, for COVID, and they still get their flu shots, the response sometimes is not the ideal response, and they tend to be a little bit more susceptible. That doesn't mean that they have no response to vaccines, and that doesn't mean that getting the flu is life-threatening, but certainly infections can be more severe or longer lasting in people who take these medications. And I think the side effect that people always tend to gives us pause, both on the side of us who are prescribed these medications and on the side of people who take them, is that there is a long-term slight risk of cancer because our immune system naturally guards against cancerous cells. And so when we suppress our immune system, the rate over time of cancerous cells getting through can be higher. Is it a lot higher? No. But it is known that over 10, 15, 20 years, there's a slight increase in the risk of cancer in transplant recipients in general. So does that impact a person's decision? For sure it does. I saw an islet candidate today who said, I don't care about that. My control is so miserable. I'd be happy to experience 15 or 20 years without having to worry about my control. Other people will say, oh, I guess my insulin control isn't that bad. I really don't want to go there. So it's a very, very personal decision based on how one's quality of life and quality of health is with their current insulin therapy.
Sean asked if you're familiar with less toxic and more targeted immunosuppression methods like, I'm probably going to mispronounce this, tegoprubart. That's a biologic. I can never pronounce those names.
We're trialing tegoprubart right now. We're trialing it in kidney recipients, and actually our center was the first enrolled the first patient in the world on tegoprubart. It works. It's very exciting. I would say the jury is still out on how well it will fit in. I don't think we're going to use it by itself, for example. And like all anti-rejection, it isn't. You can't control the immune system without somehow taking a risk of viral and bacterial infections being more burdensome. So it is very good. It has a great sort of side effect profile in terms of it doesn't, result in liver test abnormalities or kidney function abnormalities. But it is an immunosuppressive, and we're going to still be working out how it fits into the armamentarium for a little while.
And Paul asks, how long before we see islet transplant from lab-made islets with no lack of quantity and without the need for anti-rejection medications due to the islet protection methods discussed here?
Well, to give you it's a great question, and obviously, I'm pretty sure whatever I say is not quick enough. But to give you an example of how these processes work, Vertex started their trial in 2021. We are now approaching the end of the phase 1/2 trial. They will then go to a phase 3 trial where they will then enroll many more patients to assure the regulators that the protocol is safe. That will take, each of these trials involves at least a year of follow-up. And so even for something like VX-880, we're looking at, probably 5 years down the road before FDA, Health Canada, and these other agencies say, okay, there's enough safety data and efficacy data here that this is okay. It's really done for the protection of the public. It's not, it's not that, we don't want these therapies to be available right away. It's that when the regulators give the green light, that means this product is good for widespread consumption.
I just want to also mention that my colleague Laura has been answering some of the questions in the chat. So if you've been getting a response from Laura Green, she is our JDF staff member who is our resident expert on cell therapy, which is why she's been helping with the chat. So I just wanted to recognize and acknowledge her as well in case you've been getting answers from her. I think we have time for one more. And there's a question about iPSC, which I will admit I don't know what that is, but
induced pluripotent stem cells.
Okay. She just asked if we'd already discussed it. So
yeah, that's what is being used. Pardon me, embryonic stem cells are being used in the Vertex product, and again, the iPSC approach, which is what was used in the Chinese report, is slightly different, but the product at the end of the day is a stable, mature beta cell that functions in a normal way, and that's not a minor requirement. You want to produce a cell that produces that makes the right amount of insulin to the right glucose stimulus. You don't want something that makes a lot of insulin to a little bit of glucose. That would be dangerous. And so the fact that these cells, both in the Chinese study and the embryonic stem cell derived islets from Vertex, the fact that they function so normally is extremely encouraging.
So I think I'm going to wrap it up now because we are basically at 9 o'clock. I put in an email address in the chat, research@jdrf.ca. If you have any further questions you want us to address, please send anything to that email. We will address them tomorrow. This presentation will be sent out to everybody by email tomorrow. It will also be posted to our YouTube channel. But I really, really wanted to thank you so much, Dr. Pereski. I don't know if you know this, but he also did this presentation in French. the hour before this. So he has been talking in 2 languages for the last 2 hours about this very important field. He is also, as you can see from the title that I read earlier, probably one of the busiest men in this country. So we're so grateful to you for your expertise and your time tonight in 2 languages. And I think I speak on behalf of everybody when I say thank you for sharing, but thank you for what you're doing as well, because I think it's bringing a lot of hope to people on this call, and hope is an important thing.
Well, thanks to what JDRF is doing, quite honestly, because the support for these activities across the country is essential. I mean, these are amazing advances that we're seeing in other labs. I'm kind of more of a user of these new developments, but the advances are truly amazing, cannot be done without the support of funding agencies like JDRF and the support of the public. So thank you very much.
Thank you. And with that, I will wish everybody a really good evening. And we hope you'll join us next month for our last one of the year. And thank you again, Dr. Paraskevas.
Bye, my pleasure.